but it is my great pleasure to introduce our first speaker today, Dr. Joanna Achinger-Kawecka. Sorry if I mispronounced that. But anyways, Joanna did her PhD at the University of Tübingen in Germany, where she was awarded the Marie Curie Fellowship. In her PhD work, she established a novel predictive microRNA signature for endocrine-resistant breast cancer. From there, she went on to do her postdoc at the Garvan Institute in Sydney, Australia, in the lab of Professor Susan Clark. For those of you who know your time zones, you'll notice that this means she is an absolute trooper for presenting in the middle of the night. She looks remarkably put together, much more so than I would. And when we were chatting before, she sounds very bright and chipper. So I guess she's a middle of the night morning person, bird. But no, so thank you so much for coming to speak. In her postdoc, she has established cutting-edge experimental and computational approaches to study the 3D genome organization. And her research is focused on investigating the role of the 3D epigenome in cancer, particularly in driving tumor progression and treatment resistance in breast cancer and prostate cancer. Today, I'm very excited to hear her work on 3D epigenome remodeling in endocrine-resistant breast cancer. So please join me in giving a very warm, if virtual, welcome to Joanna.
Thanks so much for the introduction. And yeah, thanks so much for the opportunity to present our lab's work. Yes, so as you know, the breast cancer remains a major cause of cancer-related deaths among women worldwide, and around 70% of breast cancer patients are estrogen receptor positive, and these patients will receive some kind of endocrine treatment. And as you can see on the Kaplan-Meier plot, this endocrine treatment, like tamoxifen, very significantly reduces this patient's risk of relapse. However, there's still around 30% of patients that are treated with endocrine treatment who will at some point stop responding to therapy and relapse. And that is due to the development of endocrine resistance in their tumors. And our lab has a very long-standing interest into understanding what are the exact mechanisms of endocrine resistance development. And we focus on the epigenome and its role there. So in order to study the role of the epigenome in endocrine resistance, we use an established and very well characterized by now cell line model. So in this cell line model, we have ER-positive breast cancer cell lines, MCF7s, and then we either treat them for a very long time with tamoxifen until the development of tamoxifen-resistant population, which we call TAM-R, or we treat them with a different endocrine treatment, which is called fulvestrant, and then we wait until we have a population of fulvestrant-resistant cells, which we call FASRs. So using this model, a few years ago, we have shown that endocrine resistance is associated with large changes in DNA methylation. And you can see here on the example that this was mostly DNA hypermethylation. So this is just an 450K methylation array. And you can see that in MCF7 cells, this region is largely unmethylated. It's mostly at enhancer regions, and it becomes methylated in the two resistant cell lines that were studied. So as I mentioned, this hypermethylation was mostly occurring at enhancer regions. And importantly, when this hypermethylation was associated with ER binding and exactly with loss of ER binding at these regions. So based on this work, when I joined the lab, I hypothesized that this loss of ER binding at enhancer regions that is caused by this high DNA methylation will then result in a disruption of this long-range chromatin interactions or enhancer promoter interactions, and therefore will lead to change in gene expression. So to study this and establish the role of 3D chromatin organization during the development of endocrine resistance, I used in situ Hi-C in the parental MCF7 and the two resistant TAMR and FASR cell lines. And I looked at changes in 3D chromatin in relation to then histone modifications and different transcription factor binding. I also looked at DNA methylation changes, this time with a whole genome approach, so whole genome bisulfite sequencing. And we have also performed RNA-seq to look at consequences as a gene deregulation. So just shortly, we know that the genome is organized in a, like the DNA is organized in a very tightly regulated manner inside the cell nucleus. And at the level of single genes, the DNA or the chromatin is organized into enhancer promoter loops. So this loops can connect then regulatory elements like enhancers or repressors, and they can connect them to the target gene promoters, and this can facilitate correct gene expression patterns. So from our Hi-C data, we used diff-Hi-C, which is a package for calling differential interactions, and we identified around 2,000 differential interactions between the MCF7 and TAMAR cells, and around 5,000 differential interactions between the MCF7 and FASR cells. So as you can see on the PCA plot of top most variable interactions, there's very high dissimilarity between the parental and the two resistant cell lines, but there's also not many similar differential interactions between the two resistant cell lines, showing two very different modes of resistance development. So overall, we observed both loss of existing interactions as well as gain of novel interactions in the two cell lines. And there were very few common differential interactions between the TAMAR and FASR. So we had a closer look at those differential interactions. So we used chromHMM to assign these interactions to regulatory elements, and we saw that gained interactions were highly enriched for active enhancers and promoters. And considering this, that there were enhancer-promoter interactions, we obviously looked at the change in gene expression at these interactions. And overall, we saw that gained interactions resulted mainly in upregulation of gene expression for the genes that were located at the anchors, while loss of interactions was mainly associated with downregulation of gene expression. So here's a visual example of how the data looks. So you can see at the GRIP1 gene locus, in the parental endocrine sensitive MCF7 cells, there's this long range enhancer promoter interactions present. These interactions are completely lost in the two resistant cell lines, TAMAR and FASR. And then the GRIP1 gene, as you can see on the RNA-seq track, is completely silenced. And we know that low expression of GRIP1 gene is associated with poor outcome in patients treated with endocrine treatment. So one of the most differentially expressed gene in the resistant cell line, both of the cell lines, is ESR1. So this is the gene that encodes the estrogen receptor. And you can see that it's very highly downregulated in TAMAR cells, and its expression is almost completely lost in FASR cells. And estrogen receptor binding has been widely studied in the context of endocrine resistance by Jason Carroll and many others. And as you can see, the actual binding of estrogen receptor is very significantly altered in the resistant cells, with large gain and loss of ER binding. And Jason Carroll has also shown that this occurs in clinical samples from patients. So we tried to overlap the identified differential interactions with the alterations in ER binding. And we saw that lost differential interactions or loss of ER binding was mainly resulting in loss of interactions in the tamoxifen-resistant cells, while gain of ER binding was associated with gained interactions. And overall, we saw that around 30% of both lost and gained interactions were actually ER-bound differential interactions. And these ER-bound differential interactions were enriched for key breast cancer-related pathways and genes like Wnt, HER2, or estrogen receptor. So our previous work using methylation arrays has shown that there's huge hypermethylation associated with ER remodeling in those resistant cells. And here we confirmed it using whole genome bisulfite sequencing, when we again see that hypermethylated, differentially methylated regions are associated with loss of ER binding, while hypomethylated, differentially methylated regions are associated with new ectopic ER binding cells. And overall, you can see on the average profile that loss of DNA methylation occurred at ER binding sites that were gained in the resistant population. And importantly, when we acquired some primary and metastatic matched patient samples from endocrine-resistant patients, and we performed whole genome bisulfite sequencing, and we saw that at ER binding sites that were gained in endocrine-resistant tumors, we saw that there was loss of DNA methylation in the metastatic as compared to the primary tumors. Mind you that this is only 9 versus 10 patient samples, so we can't really do any proper statistics on it unless we're getting more samples. So here is, again, just a visual example of how the differential interactions match with the methylation data. So at this gene locus, you can see long loops being present in the endocrine sensitivity cells in the top, and this interaction is almost completely lost in the resistant cells. And when we looked at anchors of these long-range loops, we can see that they're enriched for ER binding in the sensitive cells. This binding is completely lost in the resistant cells. And below, you can see that we've put the methylation track for the MCF10 and TAMAR cells. And if we zoom into that methylation, it's quite clear that these anchors show increase in methylation in TAMAR versus MCF7 cells. And this increase in methylation is associated with enhancers, loss of ER binding, and can be observed across the matched primary to metastatic tumors, although there is a high variability in patients. So in our Hi-C data, we also looked at a higher order organization of the genome. So at above the interactions, the genome is then structured into compartments. So we called compartments A and compartment B from the PC1 values. So this is how the data looks on the screenshot. And overall, the genome is organized into active A compartments, which will be mainly located in the middle of the nucleus. They're associated with high level of transcription and mainly open chromatin. While inactive B compartments would be mainly located in the lamina and are associated with gene silencing and closed chromatin. So we observed around 2% of compartments that switched between the sensitive and the resistant cells, and we observed quite similar changes of A to B and B to A switches. As had been previously shown, we saw a very significant association with gene expression, where if you change from compartment active to inactive B, you saw loss of gene expression, and opposite for compartments that switch from inactive to active. Yeah. So here is an example of such a region. So you can see in the MCF7 cells, this region is mainly A compartment. That whole region becomes a B compartment in the TAMAR and FASR cells, and we can see loss of gene expression for genes located within that structure. So we obviously wanted to know how that relates now to ER binding profiles that we saw and the ER bound differential interactions. And we saw that if a compartment switches from A type to B type, we see enrichment for loss of ER binding. And if compartment switches from B type to A type, compartments are then enriched for gain of ER binding. And here is just another visual example when the whole region is a very long B compartment. In the MCF7 cells, in the TAMAR cells, you can see that there's a change in the compartment PC1 value that becomes more active, and it actually goes over and switches into an A compartment. And when we look at ER binding at this region, you can see that you get this ectopic ER binding coming out at that region. And that's at the enhancer promoter interactions for the gene. So overall, we have shown that 3D epigenome remodeling is a key mechanism that underlines endocrine resistance in ER-positive breast cancer. We found differential ER-bound enhancer promoter interactions, and these were associated with aberrant expression of key breast cancer genes. They were located at altered ER binding, and this was associated with DNA hypermethylation at their ER-bound anchors. We also saw change in A and B compartment structures, which was occurring with differentially expressed genes and altered ER binding sites. So with that, I'd like to thank my lab, my supervisor mostly, Susan Clark, Fatima, who's done a lot of those experiments, and other members of the lab and our collaborators. And thank you for your attention.
Thank you, Joanna. That was fantastic. So we've got a couple of questions already. And let's remind people, if you want to ask your question, you can either you can type them into the Q&A box, or you can raise your hand and I'll unmute you. So first one is from Eddie Rodriguez Corballo, and he asked, do you know if the association between methylation and gain and loss of contacts, is it a cause or a consequence?
So with ER, I know that CGCF, which actually causes interactions, which is an architectural protein, is methylation sensitive, and it actually affects the binding. But with ER, I'm not. So the canonical ER binding motif can get methylated, and we do see some ER binding sites that seem to be actually driven by methylation. But I do think that largely it's the ER binding remodeling then results in the hypermethylation of sites that are not being used in the cells. So consequence, but we don't have a temporal model to prove it yet.
And then related, Mike Carey asked, in the browser tracks you showed CTCF binding seems to decrease alongside the ER and the TAMAR cells. Could some of the effects be due to hypermethylation effects on CTCF binding? And this is possibly related to the compartment switching.
Yeah, so that's kind of what we are looking at now. So we have different cell models that have higher methylation, and then we demethylate them and we look at CTCF binding. And I know that Marano has been doing that in lots of other labs. Yes, but CTCF seems to be, in that context, very methylation sensitive. So it'd be very interesting to know as well.
Yeah, and then Arcana Dasarathi asks, says, fascinating talk. I was curious if GATA3 occupancy changes in your system. Is there some data suggesting it helps ER alpha binding through enhancer accessibility?
Yeah, so we actually looked at motifs for what most motifs were actually located at the anchors of differential interactions. And we saw, except for ER, we also saw FOXA1, GATA3, we saw CTCF, obviously, because they're interactions. I think there was some GATA3 that data for the MCF7 cells that I looked at, but we didn't have the matched resistance, so we couldn't tell if it's actually going to change. But yeah, so very interested pioneer factors, yeah. Cool.
Yeah, and then Satyanaran Rao asks, is chromatin opening by FOXA1 affected by DNA methylation? And how does FOXA1 differ in the MCF7 versus TAMAR cell lines?
Yeah, so Jason Cowell has a lot of studies on FOXA1. FOXA1, I would say that always just has to bind first before ER can go on the chromatin, and they usually bind together. I don't think we had FOXA1 when we looked at this data. We now have a different model when we treat the breast cancer cells with epigenetic therapy to demethyl the binding. And we actually can see that when you increase ER binding on the chromatin, you first have the FOXA1 coming there and opening the chromatin. Some ATAC-seq would be nice too, to show that you can actually open the chromatin before the FOXA1 or after FOXA1.
Okay, and then we'll maybe go one more question before wrapping up. And this is from Alan Underhill, who asks, do any of the methylation changes coincide with activating mutations in ESR1?
So the mutations are very interesting, but none of the cells that we have ESR1 mutations. We now have some PDX models for our collaborator that have the tumors have that ESR1 mutation. I don't know what would be the effect on DNA methylation really, because the ER will be just active all the time. But we're looking at studying that in the future with the PDX models, more like the cell lines.
Awesome. Okay, so we'll leave it there. I'll note that there is a coffee session afterwards, and so if your question wasn't answered, you can join us in the coffee session and ask Joanna your question there. Or Joanna, if you want, you can read through the chat and the Q&A and answer the questions by text. But thank you so much for a fantastic talk.